Hi, everyone, and welcome to our third Infogest Viva Webinar. Talita and I would like to introduce today's speaker, Dr. Raquel Sousa, who has recently completed her PhD at Brodkorb in Switzerland. The title of our talk is Development of an in vitro digestion protocol for assessing protein quality and functionality. The method that Raquel developed will have a high impact because, in contrast to other methods, it will allow to assess the overall digestibility of proteins in vitro and put actual numbers on it. So this is increasingly important and is especially relevant now for the study of novel or alternative proteins as they are becoming available. So Raquel, the floor is yours.
Hi, good afternoon. It's a pleasure so that I can present my work in this webinar and spread it around to the most people possible. So thank you very much for the opportunity, and I hope you enjoy the talk. I will be happy to answer all the questions at the end.
Thank you, Raquel. So Talita is going to share your.
Good afternoon, my name is Raquel Sousa, and today I will present you the work I was developing during my PhD thesis at Brodkorb, which consisted in the development of an in vitro digestion protocol for assessing protein quality and functionality. This work was supervised by Lotte Egger, Reto Portman, and Isidra Recio. The gastrointestinal system consists of the oral cavity, esophagus, stomach, small intestine, which is divided into duodenum, jejunum, and ileum, large intestine, and the assisting organs such as salivary glands, liver, gallbladder, and pancreas. It has as fundamental function to mechanically, chemically, and enzymatically disintegrate food into nutrients, the absorption of these nutrients, and the excretion of the waste products. The digestion starts already in the mouth, where by chewing, food is broken into smaller pieces that are lubricated with saliva, forming a semi-solid mass, the so-called bolus, that can be swallowed. Carbohydrates and fat digestion starts here in the mouth by action of the salivary amylase and lingual lipase. The bolus follows then to the stomach, where the gastric contractions keep on breaking down the food particles to sizes of 1 to 2 millimeters, forming the semi-liquid fluid called chyme. In the stomach, the very acidic pH favors protein denaturation, which are then hydrolyzed by pepsins, and gastric lipase continues fat digestion. As the chyme enters the small intestine, pancreatic enzymes, which are a complex mixture of proteases, amylases, and lipases, are released. Fat is hydrolyzed by pancreatic lipase, and carbohydrate digestion continues with pancreatic amylase. Proteins are first attacked by endopeptidases such as trypsin, chymotrypsin, and elastase, and then by exopeptidases. Essential all protein is absorbed in the duodenum or proximal jejunum in the form of D and 3 peptides or 3 amino acids. However, after the action of pancreatic proteases, protein hydrolysis yields 30% of 3 amino acids and 70% of oligopeptides. The intestinal epithelium is covered by tiny projections called villi and macrovilli, which further increase the total area for absorption and host the brush border enzymes. The brush border enzymes are responsible to reduce poly and oligopeptides to 3 amino acids and D and 3 peptides that can be absorbed into the bloodstream. The intestinal epithelium is produced by stem cells that can differentiate into absorptive and secretory cell types, including the rare hormone-producing enteroendocrine cells, which express different receptors and transporters on their surface for nutrient sensing. When sensing nutrients, this cell releases a battery of gastrointestinal hormones. These hormones, such as CCK and GLP-1, among others, modulate the digestive process, food intake, and glucose homeostasis. Together with other nutrients, protein has a potent effect on activation of these receptors, triggering the release of hormones. The interest in gut peptides has increased in the recent decades, with the finding that they have profound and sustained physiological effects on appetite and insulin release. Therefore, formulating foods which contain target nutraceuticals represents another possible approach to treat obesity. Protein is an essential nutrient that can be found in the wide range of foods such as meat, fish, dairy, nuts, and pulses. Human body doesn't have reserves of proteins, and therefore it is necessary to ingest enough protein to ensure that processes such as growth and maintenance are not compromised. From enzymes to hormones, muscles, and tissues, protein can be virtually found everywhere in the human body. And despite the huge variety of existing proteins, they are all made from the same 20 basic building blocks called amino acids. The amino acids can be divided into essential and non-essential. The essential amino acids are the ones our body cannot synthesize, and therefore they must come from foods, whereas the non-essential can be synthesized by our cells. Dietary protein can be defined as complete proteins when they have all the essential amino acids, which is the case of most of the animal proteins except collagen, or as incomplete proteins in the case they lack one or more essential amino acids, which is the case for most of the plant proteins except soy. Although present in our diet since ever, animal proteins are becoming more and more unpopular. The greenhouse gases emission, pollution of the fresh and marine waters, and deforestation associated to animal protein production lead to a great movement towards reducing animal protein consumption, especially meat, which is also associated to human health problems. In the recent years, many types of alternative meat made from grains, legumes, or fungi have been launched on the market. However, as I mentioned before, plant proteins are normally incomplete protein sources, and therefore there are some concerns that vegans or diets containing mainly plant proteins are nutrient deficient. Therefore, it's very important to determine protein quality accurately. There are different methods for evaluating protein quality. Protein efficiency ratio, protein digestibility corrected amino acid score, and digestible indispensable amino acid score are among the most common. Protein efficiency ratio is determined in rats that are fed with a test diet and a reference diet, and then the ratio of weight gain of both diets is calculated. However, this method is easy to perform and calculate. It implies animal use, more precisely rats, which have different needs and anatomy from humans. In 1993, FAO proposed the protein digestible corrected amino acid score. It is calculated by quantifying the amino acids present in the tested food. Then, if we take lysine as an example, we divide the amount of lysine present per gram of food protein by the amount of lysine in the reference protein, and then multiply by the fecal true protein digestibility. However, this method has some disadvantages, such as the use of fecal digestibility that can lead to overestimations of protein digestibility, the assumption that all amino acids have the same digestibility as crude protein, and values truncated to 100, not allowing the evaluation of the nutritional significance of protein as part of mixed diets. To address these issues, in 2013, FAO proposed a digestible indispensable amino acid score. The calculation is basically the same as the PDCAS, except here, ileal digestibility of individual amino acids is used instead of fecal crude protein digestibility. This methodology allows a more accurate determination of protein quality because the samples are collected right where they are absorbed before entering the large intestine and being modified by the microbiota. However, sample collection at ileum level is very invasive and therefore limited data is currently available, not allowing the full implementation of this method. Ileal digestibility can be calculated by taking samples from nasoileal intubated healthy volunteers, which have a tube introduced from their nose down to the ileum where samples are collected, or from ileostomized patients, which have their small intestine exteriorized and the ileal digesta can be easily collected into a stomach punch. However, it is very hard to find ileostomized patients, and the nasoileal intubation is very invasive, and therefore not that many people are willing to participate in this kind of studies. Together with this, human studies are costly and loaded with ethical issues. Recently, cannulated pigs have been validated as a model to replace human assays. Although more accepted and easy to perform when compared with human assays, pig studies are still time-consuming, costly, and facing ethical issues. Therefore, FAO promotes the development and validation of in vitro methods to predict amino acid digestibility and bioavailability in humans. In 2014, Infogest Cost Action developed an in vitro static digestion model based on physiological relevant conditions, which is composed by three different phases: oral phase containing amylase, gastric phase containing pepsin and gastric lipase, and intestinal phase containing pancreatic enzymes and bile salts. This protocol has been tested for its reproducibility and biological relevance by an interlaboratory trial and pig assay respectively, which proved that the in vitro protein hydrolysis is a good approximation to the in vivo situation. Having this protocol as a starting point, we hypothesized that it could be possible to determine dias in vitro just by simply replacing the ileal digestibility of the individual amino acids by the in vitro digestibility. Taking again lysine as an example, the in vitro digestible indispensable amino acid ratio would be then the amount of lysine per gram of food protein multiplied by the in vitro digestibility of lysine, what we call the dia measured, divided by the amount of lysine present in the reference protein. The digestible indispensable amino acid score is then the lowest dia and it's reported as the limiting amino acid. Therefore, our main goal was to develop and validate with in vivo data an in vitro digestion method able to assess protein quality in order to reduce the need of in vivo assays. For that, we established 4 milestones. First, we characterize and test protein hydrolysis of selected protein sources with the Infogest harmonized in vitro digestion method. Second, we amended the Infogest in vitro digestion protocol in order to develop an analytical workflow to assess protein digestibility and protein quality and its comparability with in vivo data. We then applied the in vitro digestibility analytical workflow to evaluate the protein quality of highly transformed plant-based protein sources. And as last, we evaluate the effect on in vitro digestion products on satiety hormone release by STC1 cells. Right now, I will start showing you the results of the characterization of the selected protein sources and the protein hydrolysis after digestion with the Infogest static protocol. The products used in this work were the same products selected for the Proteus project, where they were determining dias values in pigs and humans in order to validate the pig as a replacement for human assays. They were selected in order to reflect protein sources used in diets around the globe. Both animal and plant proteins, complete foods and isolated proteins were selected with expected different qualities and different digestibilities. The selected protein sources were sorghum, all-grain cereals, black beans, pigeon peas, peanuts, collagen, whey protein, and zein. The protein sources were characterized for their macronutrient composition and the major proteins present in each substrate were identified with LCMS after tryptophan gel digestion and the amino acid profile by HPLC. All the protein sources were digested according with the Infogest static protocol, and after digesting, they were centrifuged and separated into supernatant and pellet. Protein degradation at the end of every digestive phase was evaluated by SDS page, and in this slide I have depicted two examples, the whey protein and black beans. Samples from undigested products, oral phase, gastric phase, and intestinal phase are here represented. At the end of the gastric phase, there were still some intact proteins such as beta-lactoglobulin and phaseolin. However, at the end of the intestinal phase, it was only possible to identify proteins coming from pancreatic enzymes. The peptide profile at the end of each digestive phase was also studied with LCMS, and here we see the peptide profile of the beta-lactoglobulin on the left and phaseolin on the right. These graphs represent the peptides generating during the different digestive phases, oral, gastric, and intestinal, and they are color-coded according with the abundance of the identified amino acids, where red represents the higher number of identified amino acids, blue the lower number of identified amino acids, and white represents the absence of identified amino acids within the protein sequences, which is represented by the x-axis of the graph. It is possible to see an increase of peptides from the oral to the gastric phase, indicating the degradation of the intact protein into peptides, and a decrease from the gastric to the intestinal, indicating a degradation of the peptides that were generating during the gastric phase into free amino acids. The formation of free amino acids and short peptides was also evaluated by OPA method, and looking at the graph, we can see that the protocol allows seeing difference in protein hydrolysis. However, the assessment of total digestibility was not possible due to the high enzymatic background. Therefore, some protocol amendments as well as the development of an analytical workflow in order to assess protein quality were needed. We started our experiment with a protein input of 40 milligrams, which was the protein input used in the pig assay that validated the Infogest protocol, and we realized that the substrate enzyme ratio was very low, with the enzymes contributing to 2/3 of the nitrogen in the system, originating a high enzymatic background. Therefore, we digested different amounts of proteins, 40 milligrams, 60 milligrams, and 80 milligrams, using the same amount of enzymes, and we saw a decrease in protein digestibility with an increase of protein input. This decrease was not visible for whey protein, which is a highly digestible protein. However, it was very well visible for zein, peanut, and all bran. Like that, we conclude that there were not enough enzymes to guarantee the complete hydrolysis of 60 and 80 milligrams of protein, and that enzymes need to be in excess in order to see differences in digestibility. The next issue to address was the solubilization of the pancreatine in the simulated intestinal fluid. In the first experiments, we realized that the pancreatine was not well dissolved, originating a slurry rather than the homogeneous solution, and it was very hard to pipe it in a reproducible and representative way, leading to inconsistent results between replicates. Therefore, we tried to solve this issue by dissolving the requested amount of pancreatine in the simulated intestinal fluid, vortexing for 10 seconds, ultrasonicating for 5 minutes, followed by 5-minute centrifugation. The supernatant was collected and the trypsin activity was measured. We didn't find significant differences in trypsin activity, and therefore, from that moment on, all the digestions were performed with the supernatant of the treated pancreatine, and we saw a huge improvement in repeatability of the results. At the beginning of our study, we were using a water-blank digest to deduce as enzymatic background. However, we realized that there was a high enzymatic autodigestion of the enzymes in the absence of any kind of substrate, leading to underestimation of the protein hydrolysis. In vivo assays use a protein-free meal in order to quantify the contribution of endogenous protein. In this study over here, they use a protein-free cookie, and therefore we decide to bake the same cookie with the very same composition depicted here in this table and use it as our control. In every set of digestion, a protein-free cookie was digested in parallel with the tested protein sources. In order to calculate the in vitro dias, we had first to decide what would correspond to the bioavailable fraction of the in vitro digest. Our model is a static model where at the end of the digestion, both digested and undigested fractions are together inside the same reaction vessel. Besides that, our model lacks the Bresch border enzymes, which are responsible for the final stage of the protein degradation by converting polypeptides into D and 3 peptides and free amino acids. Therefore, we decide that the in vitro bioavailable fraction would be composed by free amino acids and peptides up to 10 amino acids, which in vivo would be further degraded by the Bresch border enzymes and becoming available for absorption. But as I said, everything was inside the same reaction vessel, and in order to separate these two fractions, we decided to perform some precipitations. Different precipitation agents were tested, and the resulting soluble fraction was injected in a size exclusion chromatography column in order to check the size distribution of the molecules present in this fraction. Precipitation agents like perchloric acid, acetone, TCA, tannic acid, methanol, among others, were tested, and methanol 80% proved to be the best condition by yielding a best recovery and due to the fact that it's easy to evaporate without leaving any traces in the samples. In order to characterize the size of the molecules present in the fractions resulting from the methanol precipitation, we had to calibrate the SEC system. For that, several peptides with known sequence and molecular weight were injected. In this technique, the heavier molecules elute first and the small molecules elute later. By drawing a logarithmic regression between the retention time and the molecular weight, we could determine which would be the elution time corresponding to the 10 amino acids we defined as a limit for the in vitro bioavailable fraction, and this was approximately around 40 minutes. All the tested proteins were digested, precipitated with methanol, and the fractions resulting from this precipitation were injected in a SED column. Here I show a few examples, and we see that the majority of the peaks from the pellet elute before 40 minutes, whereas the majority of the peaks from the supernatant elute after 40 minutes, proving that the methanol 80% condition is a very good condition to isolate the in vitro bioavailable fraction from the whole digesta. Surprisingly, we found a very intense peak present in the cookie digest that was not present in the other tested protein sources. So, in order to discard the possibility that this peak could have protein origin, we injected in parallel with the cookie digest a water blank containing the same type and amount of enzymes, and we saw that the peak that was present in the cookie was not visible on the water digest, proving that this was not from protein origin. Analytical workflow in order to calculate the in vitro digestibility, which is represented here in this slide. We first digested all the investigated protein sources in parallel with the protein-free cookie. We precipitate every digest with the methanol in order to obtain the bioavailable fraction, which would be the supernatant, and the non-bioavailable fraction, which is the pellet. And both fractions were analyzed by Kjeldahl, OPA, and HPLC in order to determine the total nitrogen, primary amines, and the individual amino acids respectively. For all the analytics, the samples were hydrolyzed first in order to break down all the peptidic bonds. The in vitro digestibility was then calculated by dividing the digestible fraction by the total of the food. After the first calculations and consequently comparison with the in vivo results, we realized that we were having higher digestibilities for the isolated proteins when compared with the in vivo results. At this moment, We get to know that in the human assay, the isolated proteins were given together with the protein-free cookie and orange juice and mixed with fat, carbohydrates, sugar, and other nutrients in the pig trials. We then decide to perform the digestion of the isolated proteins together with different amounts of protein-free cookie, and we saw a decrease in digestibility with the incorporation of cookie in the digest. This decrease was more pronounced for zein, but not so much for whey protein and collagen. We could get very near to the in vivo results for zein and whey protein, however, collagen was still very far off. And this can be because collagen solubilization is reported to be strongly dependent on the pH, and in the in vitro system, the gastric pH is directly adjusted to 3, while in the in vivo situation, the decrease in pH is gradual. Here I show the in vitro total digestibilities calculated using the 3 different analytical endpoints, TN represented with the black bars, OPA represented with the gray bars, and total amino acids represented with the light gray bars compared with in vivo data. Black beans, pigeon peas, all bran, whey protein, zein, and collagen were compared with the average human pig. However, peanut was abandoned from the project, so we didn't have this value for pig and human, and we did a comparison with rat data available in literature. We can see that the 3 different analytical endpoints give very similar results. When comparing the in vitro digestibilities with in vivo data, we can see that the in vitro values are slightly higher than the in vivo ones. Here I show a Bland-Altman graph comparing the individual amino acid digestibility obtained by the in vivo and in vitro systems. In the y-axis is the difference between the 2 methods, and in the x-axis it's the average of the 2 methods. The middle line is the bias between the methods, and the upper and lower limits are plus minus 2 standard deviations. Because more than 95% of the values are within these 2 limits and the bias is 1.2%, we can conclude that these 2 methods are comparable. We then calculated the in vitro DIAR, as I explained before, and compared with the in vivo DIAR values. The black bars are the in vitro values, and the white and gray bars are the in vivo values. We see a nice agreement between the results obtained by both systems and even the same prediction for the limiting amino acids. There are no bars for the in vivo DIAR values of sulfur-containing amino acids because the in vivo cysteine values were not reported. Here is presented the correlation curve between the in vivo DIAR values present in the y-axis and in vitro DIAR values in the x-axis. We obtained a slope of 0.95, very close to the idyllic slope of 1, and an R value of 0.89. Again, the Bland-Altman comparison, and only 4 values are outside the upper and lower limits, and the bias is 0.1%, indicating an extreme similarity between the 2 different methods. We then calculated what we call the proxy in vitro DIAR, which is calculated the same way as the DIAR, but instead of multiplying the individual amino acid digestibility, it is multiplied by the in vitro total digestibility obtained by Kjeldahl or OPA. We correlate the proxy in vitro DIAR with the in vivo DIAR, and in this graph, it's possible to see a good correlation between the 2 results. Like this, and in the case of the total amino acids analysis is not available, proxy DIAR can be used as a good approximation based on total digestibility. In order to test the applicability of the newly developed methodology to highly transformed foods, different plant-based protein sources were tested. 2 veggie burgers, one based on soy protein and another one based on the mixture of pea and fava protein, and also a soy-based meat analog, and all the ingredients were compared with beef burger. All the tested protein sources were analyzed for their macronutrient composition, as we can see here in this table. And in this graph on the right-hand side, we can see that the plant-based protein sources have a well-balanced amino acid profile. We then calculated total digestibilities, as I explained before, and in this graph, we have the total digestibility calculated based in the Kjeldal results represented in yellow, total amino acids in green, and OPA in blue. And as we can see, all the digestibility values are over 70%, indicating a good protein digestibility for all the tested products. The impact of grilling on the amino acid digestibility was evaluated, and in this graph, the dark bars stand for the raw burgers, while the bright bars stand for the grilled burgers, and we can see that the P-FABA burger amino acid digestibility is negatively affected by grilling. However, this impact is not visible for soy and beef burgers. The impact of grilling on in vitro DIR values was also evaluated, and a positive effect was seen for the beef burgers. However, it was not clear for the vegan burgers. Still, we can conclude out of this graph that plant-based burgers are high-quality protein sources because the DIR values are between 75 and 99%, and as expected, beef burger is an excellent quality protein source because the DIR values are all above 100%. In order to somehow validate our results, we compared our in vitro DIR values with in vivo DIR values found in literature, and we can see in these graphs that both systems predict the same limiting amino acids, yet overall, the in vitro values are slightly higher than the in vivo. Nevertheless, we should keep in mind that these are not the same exact products, and protein isolates can vary a lot. At last, the effect of the in vitro digestion products on satiety hormone released by STC1 cells was also evaluated. STC1 cells are a murine intestinal secreting tumor cell line that is routinely used in hormone secretion studies. For this study, the digestions were performed with 2.5 millimole per liter of bile salts instead of the recommended 10 millimole per liter due to cell toxicity. After digestion, the digests were split into soluble and insoluble fraction by centrifugation, and the soluble fractions were incubated for 2 hours and a half with the cells at 2 different concentrations, 2 mg protein per milliliter and 1 mg protein per milliliter. Cell viability was measured with Alamar Blue, and we saw a significant decrease for some substrates at the concentration of 2 mg protein per milliliter, and therefore we continued the experiment with a lower concentration. We then incubated digest with the cells for 2 hours and measured the hormone secretion with ELISA, and we found a dependence on the digestive phase. Hormonal secretion was higher for intestinal fractions and significant for CCK, but not for GLP-1. Looking into more detail, when comparing the hormone release for the 2 different tested concentrations, we see a significant secretion for CCK and GLP-1 in a dose-dependent manner. Besides that, we also found a very interesting pattern where the gastric digests inducing the maximal release of CCK are the same inducing the maximal release of GLP-1, and this same pattern is also visible for the intestinal digests. We then investigate the correlation between hormone secretion and macronutrients, and as expected, we found a significant correlation between CCK release and the protein content, and a significant correlation between GLP-1 secretion and carbohydrates content. CCK and GLP-1 mRNA levels were also determined by RT-qPCR after 2 hours of incubation. No dose-response effect on CCK mRNA levels for both digestive phases was found, but the dose response in GLP-1 mRNA levels for the intestinal fractions was visible. We then decide to synthesize a few peptides present in the in vitro digests of zein and collagen. The selection of these peptides was based in their high abundance in the digests, hydrophobicity, and the presence of aromatic amino acids. In this table, we see the protein from where the peptide is coming from, the sequence of the peptide, the length, hydrophobicity, and the theoretic isoelectric point. Hormone secretion from STC1 cell line appears to be dependent on the rise in cytoplasmatic calcium, and therefore, the synthetic peptides were tested for their ability to activate the cells as a screening for future hormone secretion studies. In the graph, we see that the majority of the peptides induce an increase in intracellular calcium at different levels. When ordering the peptides by source and length, shorter peptides on the left, bigger peptides on the right, we see not a length-dependent response, but rather a sequence dependency. The cell activation was more intense for the peptides P3, P8, and P1, when P3 and P8 are highly hydrophobic, whereas P1 contains a phosphorylated serine and it's very hydrophilic. To conclude, a new workflow based on the static infogest protocol was developed, allowing the assessment of protein digestibility and protein quality using DIAS as recommended by FAO. Similar protein digestibility was obtained with total nitrogen, primary amines, and total amino acids analysis. In vitro digestibility and in vitro DIAR values were highly correlated with in vivo data for the 7 tested substrates, showing that the new workflow could present a powerful tool to predict in vivo data. The developed method was applicable to assess protein quality in highly processed foods, such as plant-based burgers and meat analogs. According to the in vitro results, plant-based burgers can be considered as high-quality protein sources with highest values between 75% and 99%. Gastric and intestinal digests induced anorexigenic hormone secretion and expression in STC1 cells dependent on substrate and digestive phase. And CCK release was correlated with the increase in protein content, whereas GLP-1 secretion was correlated with increased carbohydrate contents in the digest fractions. At last, I would like to thank you all for your attention, and I would be very happy to answer all your questions.
Raquel, thank you very much for your presentation. It was really good. Now we have some questions, so let's go to the first one. The first question is, you said that the increase of protein resulted in the decrease of digestibility. But what would be your opinion, according to the human body, that it is expected that to produce more enzymes depend on the type of food that is consumed? Do you think it will be the same? How?
I mean, to transpose that for an in vitro system would be very complicated. And actually, the goal is to develop a protocol that we can use under, let's say, all the circumstances and not to have to adapt all the time. And this was actually the best solution, is like to have the enzymes in excess, and like that we can always guarantee that the protein hydrolysis occurs like fully. But yeah, like in vivo, that's what happens. Like when we ingest more food, we have more enzymes, and there is this balance. But in vitro, that would be very hard to achieve, and because we want to have a single protocol that can be used in different situations. Yeah, the solution is to have the enzymes in excess.
Okay, thank you. The next one was, how did you determine the composition of cookie? Did you do any tests before, or it was from another work? How did you choose how much of lipids and the other ingredients?
The composition of the cookie we used as the enzymatic background, it was taken from this paper from Paul Morgan that I showed in the presentation. And we just basically copied the recipe and we bake our own cookies. But recently, we have done a few experiments to change this cookie recipe, and we see that it's not really working out with other ingredients. So maybe there is a reason why they show this this formulation.
Perfect. Thank you. Let's see, we have another one here. Thank you. Very interesting. It is from Mattia. Probably I missed it, but could you explain how did you measure intracellular calcium?
This is just a screening method. We just do we just incubate the peptides, the synthetic peptides with the cells, and then we just measure the calcium that is present in the supernatant outside.
Okay. The other question is, according to the, it's about the 8% methanol. They are asking how, if you have suggestions how to apply it to the semi-dynamic methods, because in the semi-dynamic we have a big amount of samples, so we wouldn't use a big amount of methanol. If you have any suggestion.
Yeah, I mean, the protocol for assessing protein digestibility, protein quality was designed for the static protocol, not for the dynamic. And to assess protein quality with the dynamic protocol, that would be very complicated because there are different fractions, and to calculate that back, so it would be very complicated. So I don't really think that would be doable or easy to do that with the dynamic. Sorry.
Thank you very much. And what would be the next steps of the project?
So as I said, we are now testing different formulations of the cookie because there were a few comments about the margarine used for the cookie baking, and we were testing with oil and with different amounts of oil as well to reduce the amount of lipids in the cookie composition. And in the future we'll be testing different recipes for the cookie, try to optimize this composition, and also to expand the scope of the foods, because so far we just validated 7 different foods within vivo data. We would have to have much more. And now we are also talking with the Proteus project again to see if we can collaborate once more with them, because they are in the second round of the project. And it would be to validate the protocol with more foods, different ones, and to optimize the cookie recipe.
Perfect. Another one here, let me see. Lehen from the Technion Israel. It was a fascinating lecture. Thank you for the lecture. I wanted to know if you thought about using clean enzymes for digestion, then the dirty enzyme pancreatin, and thus reduce perhaps background noise.
Yeah, I mean, the protocol itself, it suggests also the use of the individual enzymes. The problem is that the individual enzymes are much more expensive, and the use of the mixture of the pancreatin, in this case, it's much more physiological. So these are the 2 reasons. It's much more physiological, and on the other hand, is cheaper. Because even though the protocol itself is quite expensive if we need to use amylase and so on, so it's trying to reduce the costs, the max.
Okay. I think we still have time for one more. Could you tell us what were the biggest challenges you had to reach your goals? Because it was a big project, I guess you had a lot.
Yeah. First, it was to find which one would be the best enzymatic background to deduce, because we were trying with water, with the enzymes mixed with the water. And after 0 minutes of incubation, after 1 hour, after 2 hours, and we tried different kinds of things. Then we found the cookie. We could find we decided, okay, let's use, but then we had the pancreatine also. So we also had to improvise with the sonication and centrifugation. And then also the amount of protein input in the system was also a bit tricky. And then to find the precipitation method that could be more efficient and easy to handle. Yeah, so I would say that was the pancreatine solubilization, the enzymatic background, the substrate input, and the precipitation. And then, okay, all the calculations behind that are massive.
Yeah, you had a lot of work.
Yeah.
So, the last one that we have here, and we still have time. In your opinion, as we can see, it is a very important protocol. So what would be your opinion for the future of this protocol?
Right now, we were trying to standardize the protocol within ISO-IDF. And if we are successful, hopefully the protocol will be used worldwide. And that's actually our goal, is that it can be used everywhere for everyone. It can also help the researchers that are working in food science, because, yeah, this is actually what was missing until now, because protein was actually the only macronutrient that was assessed in vivo. And this protocol, even though not perfect, it can be used as a screening tool, and it can be a great help for the food research, I believe.
It will be. So I think we don't have questions anymore. Thank you very much. You did a really good job. I'm sure it will be a very important protocol in our near future. Thank you very much.
Thank you. Thank you very much.
Thank you for being here with us today.
Thank you.
I also wanted to thank everybody who was here watching us. Also, would like to thank Andre, Shannon, and Gaetan, who helped us to organize the webinars. And I also wanted to announce our next speaker that will be in January. Let me share the screen. Yeah. Share. So our next webinar will be in January, and we will have a presentation with Judith Termazzi. So don't forget to register. It is a different link, so please use this one. And the next ones in February and March will be with Huoshuan Deng and Elise Charton. So the information will be announced on this soon. And thank you very much for being here. Have a happy holiday season, and see you next year. Thank you.